Shri Rajalakshmi Prakashan published. A textbook on fundamentals of agronomy 1. Authors: Dr. D. N. Jagtap, Dr. P. B. Pawar, Dr. U. V. Mahadkar. Chapter 4. Seed, its definition, characteristics of quality seed, seed treatment and its objectives, seed dormancy, causes of seed dormancy and multiplication, stages of seed. Plants reproduce sexually by seeds and asexually by vegetative parts. Grains, which are used for multiplication, are called seeds, while those used for human or animal consumption are called grains. Good stocks of planting materials are basic to profitable crop production. The seed or planting material largely determines the quality and quantity of the produce. A good seed or stock of planting material is genetically satisfactory and true to type, fully developed and free from contamination, deformities, diseases and pests. Seed is a fertilized ripened ovule consisting of three main parts, namely seed coat, endosperm and embryo, which in due course gives rise to a new plant. Endosperm is the storage organ for food substance that nourishes the embryo during its development. Seed coat is the outer cover that protects or shields the embryo and endosperm. Characteristics of good seeds. A good quality seed should possess the following characteristics. Seed must be true to its type, that is, genetically pure, free from admixtures, and should belong to the proper variety or strain of the crop, and their duration should be according to agroclimate and cropping system of the locality. Seed should be pure, viable, vigorous, and have high yielding potential. Seed should be free from seed-borne diseases and pest infection. Seed should be clean, free from weed seeds or any inert materials. Seed should be in whole and not broken or damaged, crushed or peeled off, half-filled and half-rotten. Seed should meet the prescribed uniform size and weight. Seed should be as fresh as possible or of the proper age. Seed should contain optimum amount of moisture, 8 to 12%. Seed should have high germination percentage, more than 80%. Seed should germinate rapidly and uniformly when sown. Advantages of using good quality seeds. The following are the advantages of using good quality seeds. Reduced cost of cleaning, standardization and disinfections. Uniform germination, thus avoiding replanting, gap filling. Vigorous seedling growth, which reduces weed and disease damages. Uniform growth stages, maturity and products. Maintain good quality under storage conditions. Reduced cost. Seed germination. Germination is a protrusion of radicle or seedling emergence. Germination results in rupture of the seed coat and emergence of seedling from embryonic axis. Factors affecting germination are soil, environment, water, temperature, light, atmospheric gases and exogenous chemicals required for germination of seeds. Factors affecting germination. Soil, soil type, texture, structure and microorganism greatly influence the seed germination. Environment. Generally, the environmental conditions favoring growth of seedling also favors germination. Germination does not occur until the seeds attain physiological maturity. Water, soil moisture and seed moisture imbibitions of water is the prerequisite process for germination. Both living and dead seeds imbibe water and swell. Dead seeds imbibe more water and swell rapidly as compared to good seeds. The amount imbibed is related to the chemical composition of the seed, such as proteins, mucilaginous pectins and biochemical components. Cereal grains such as maize imbibe water to approximately 1/3 of its seed weight, soybean seeds to half of its seed weight. Seed germination will be maximum when the soil moisture level is at field capacity. Slower rate of germination is noticed in places where soil moisture is near or at wilting point. Temperature. The cardinal temperature, maximum, optimum and minimum temperature for germination of some of the crops is given below. The optimum temperature is that one gives the highest germination percentage in the shortest period of time. Table: cardinal temperature for important crops. Crop match. Minimum degree Celsius 8 to 10. Optimum degree Celsius 20 to 25. Maximum degree Celsius 30 to 35. Crop rice. Minimum degree Celsius 10 to 12. Optimum degree Celsius 20 to 27. Maximum degree Celsius 30 to 32. Crop wheat. Minimum degree Celsius 3 to 5. Optimum degree Celsius 15 to 31. Maximum degree Celsius 33. Light. The most effective wavelength for promoting and inhibiting seed germination is red 660 nanometers and infrared 730 nanometers, respectively. Atmospheric gases. Most crop seeds germinate well in the ambient composition of air with 20% O2, 0.03% CO2 and 78.2% N. Exogenous chemicals. Some chemicals induce or favor quick and rapid germination. Gibberellins stimulate germination in protoplasmic seeds. Hydrogen peroxide, H2O2, is used for legumes, tomato and barley. Ethylene, C2H4, is used for stimulating groundnut germination. Seed rate. Seed rate is the quantity of seed required for sowing or planting in a unit area. The seed rate for a particular crop would depend not only on its seed size test weight, but also on its desired population, germination percentage and purity percentage of seed. It is calculated as follows. Seed rate, kg, is equal to area to be sown in square meter multiplied by test weight of the seed multiplied by 1. Divided by germination percent multiplied by purity percent multiplied by spacing meter multiplied by 1000. Seed treatment. Seed treatment is a process of application either by mixing or by coating or by soaking in solutions of chemicals or protectants with fungicidal, insecticidal, bactericidal, nematicidal or biopesticidal properties, nutrients, hormones or growth regulators, or subjected to a process of wetting and drying, or subjected to reduce, control or repel disease organisms, insects or other pests which attack seeds or seedlings growing therefrom. Seed treatment also includes control of pests when the seed is in storage and after it has been sown planted. The seed treatment is done for the following reasons: to protect from seed-borne pests and diseases, to protect from or repel birds and rodents, to supply plant nutrients, to inoculate microorganisms, to supply growth regulators, to supply selective herbicides, to break seed dormancy, to induce drought tolerance, to induce higher germination percentage, early emergence, to obtain polyploids, genetic variation, by treating with X-rays, gamma rays and colchicines, to facilitate mechanized sowing. Seed treatment and its importance. We require healthy, disease-free, and high-yielding crop. Hence there is a need of seed treatment before sowing any crop. Even though we select quality seeds, it is advised to go through seed treatment process for better germination and to prevent from seed and soil-borne diseases. Seeds can be treated with chemicals or natural bioproducts. Advantages of seed treatment. The following are major advantages of seed treatment. This process protects germinating seeds and seedlings against soil and seed-borne pests and diseases. Seed treatment improves the germination process and increases the germination percentage. Seed treatment enhances the seed viability and vigor, which are the two most important factors in agriculture or cultivation practices. Seed treatment result in early and uniform establishment and growth of the crop or plants. Seed treatment enhances nodulation in legume crops. Seed treatment is better when compared to soil and foliar application in the crop. Seed treatment results in uniform crop stand, especially in adverse situations like low moist and high moist conditions. Objectives of seed treatment. Some seeds need treatment with some specific objectives before sowing. To control disease, there are some seed-borne or soil-borne diseases. Seeds are treated with fungicides or organomercurial compounds like therum, captain, carbendazim, agrosan, cerazin, etc. Example, to control paddy blast, seed is to be treated with agrosan at 3 g per kg of seed. To have convenience in sowing, difficulties are encountered in sowing certain crops due to special characteristics of the seed like fuss of cotton seeds, coriander seeds, small seeds of chili, ragi, bajra, etc. Example, coriander seed is to be splitted by rubbing it against hard surface. Seed of chili, sesmum, bajra are mixed with fine sand or soil. To have quick germination, germination of certain leguminous crops is delayed due to thick seed coat which restricts water absorption. Such seed coats are broken to some extent by mixing them with coarse gritty sand and trampling or pounding it lightly in a mortar with a wooden pestle for breaking the thick seed coat. Sometimes seeds are soaked in water for a specified time. Example, cotton seed or paddy seed is soaked in water before actual sowing. Seed of lucerne and indigo is pounded with pestle. To increase nitrogen fixation in legumes, legume seeds are inoculated with a particular rhizobium culture. This is mixed with jaggery solution and applied to seed and dried in shade. It increases nodulation and thereby N fixation. To protect the seed against insect pests, there are some insect pests like ants, white ants, in the soil which attack on seed and eat. Sometimes, seed may be picked up by birds after sowing. To avoid this, seed is treated with repellents like camphor, kerosene or soil drenching with insecticides like BHC, heptachlor, etc. Example, the carbofuran treatment in jowar. To induce earliness, vernalization treatment. This is an important for breeding program by vernalization treatment. As a result of this, lifespan is reduced. In this, seed is soaked in water and incipient germination is induced in the form of awakening of the dormant embryo and commencing the changes favoring germination in the endosperm. Such seeds are kept in cold storage for a specified time in which germination power remains intact but the process of germination is temporarily halted. Thus, the plant spends part of its vegetative period or phase in the form of sprouted seed and the seed so treated as a dormant plant. The period from sowing to flowering is thus greatly reduced, and with such adjustment, a variety which is normally a long-duration one can be made to flower early. To induce variation, seed is treated to induce variation in its morphological and general structure by X-ray treatment. It changes the genetical makeup and helps in selection of desired types. Example, Sonora, a wheat variety, is the result of Sonora 64 treated with gamma rays. To break dormancy, some crops are having seed dormancy in fresh harvested produce. Dormancy is the state of rest period of a seed in which it does not germinate even if all the favorable conditions are available for germination. Due to dormancy of seed, we cannot use the fresh harvested produce for sowing. It is desirable if the crop get rains at maturity. Example, groundnut varieties. This dormancy is broken by treating seed with chemicals. Example, Thieria 1% treatment to potato tubers. Seed treatment for special purpose. In vegetatively propagated crops, planting material is treated with growth-promoting hormones like colchicines, gibberellic acid, Ga, indole acetic acid, IA, Ceradix, sometime cattle urine. These promotes sprouting and growth of plant. Example, onion bulbs or potato tubers are treated with malic hydrazine, MH, for avoiding sprouting and growth of sprouts and thereby reducing losses due to sprouting. Methods of seed treatment. Dry treatment, mixing of seed with powder form of pesticides nutrients. Wet treatment, soaking of seed in pesticide nutrient solutions. Slurry treatment, dipping of seedlings in slurry. Example, rice seedlings are dipped in phosphate slurry. Pelleting, it is the coating of solid materials in sufficient quantities to make the seeds larger, heavier and to appear uniform in size for sowing with seed drills. Pelleting with pesticides as a protectant against soil organisms, soil pests and as a repellent against birds and rodents. Seed treatment in important crops. Sorghum. Therum or 300 mesh sulfur. Seed is coated in seed dressing drum or earthen pot at 3.4 gram kg seed against smut disease. Bajra. Brine solution treatment is given at 20% against eat got and to discard light and diseased seed. Paddy. Seed is treated with brine solution at 3% against blast of paddy and to discard unfilled seed. Cotton. Point A. Cow dung slurry treatment. Seed is rubbed with cow dung slurry in 1:1 proportion of dung and soil for convenience in sowing, or seed is delinted by treating the seed with conch. H to sow for 2 minutes for convenience in sowing. Point B. Seed is treated with organomercurial compound like sericin, agrocin at 3 gram or therum at 5 gram against seed-borne disease like anthracnose, coriander and garlic. Seed is rubbed to split the seed for even sowing. Small seeded crops. Small seeded crops like sesame, bajra, tobacco, etc. Seed is mixed with fine sand or soil for even sowing of seed in the field. Potato. Point A. Seed is dipped in 1% Thiourea solution for breaking the seed dormancy. Point B. Seed is dipped in streptomycin solution at 200 gram in 100 liter water for 1 hour against ring rots disease. Legume crops like mung, udid, soyabean, etc. Point A. Seed is treated with therum at 3 gram per kg seed against seed-borne disease. Point B. Seed is treated with rhizobium culture at 250 gram per 10 kg seed for N fixation and better nodulation. Sugarcane. Point A. Hot water treatment 50 degrees Celsius or hot air treatment 54 degrees Celsius is given to sets for 2 hours against grassy shoot and other diseases. Point B. Sets are treated with OMC 6% at 500 g in 100 liter water by dipping for 5 minutes against smut and increased germination. Or bavistine at 200 g in 100 liter for 5 minutes. Wheat and all seed crops. Seed is coated with therum or bavistine at 5 gram per kg. Seed against seed-borne diseases. Seed dormancy. Seed dormancy refers in seeds to failure of a viable seed to germinate even when given favorable environmental conditions. In many species can get a clue about requirements for breaking dormancy by studying the natural environment of the plant. In nature, dormancy mechanisms assure that seed will germinate at the proper time. In cultivation, have highly breed crops such as cereals, which have been bred for reduced period of dormancy so that during storage dormancy controls disappear and then seeds germinate immediately upon planting. The physiological inability of a seed to germinate even under favorable condition is called seed dormancy. Many kinds of seeds can germinate immediately after maturation. The seeds in some species of citrus may germinate in the fruit. But in some cases seed passes a period of rest or inactivity before germination. Such seeds do not germinate even in favorable external conditions. Something blocks the process's germination. It is seed dormancy. Causes of seed dormancy. The following are causes of seed dormancy. Point A. Hard seed coat. Seeds of many species possess hard seed coat. Most of these seeds belong to the family Leguminosae and Malvaceae. Such seeds remain dormant. Hard seed coat prevents germination due to following reasons. Hard seed coat prevents the entry of water into the seed. Hard coat obstruct exchange of gases, especially oxygen. Oxygen is necessary for respiration. Hard seed coat causes mechanical resistance. Thus radicle does not come out. Point B. Immature embryo. The embryo is fully developed inside the seed in most of the plants before shading from the parent plant. But development of the embryo is incomplete at dispersal in few cases like Fraxinus and anemone. Such seeds remain dormant and fail to germinate. Such seeds imbibe water and the development of the embryo is completed in a few weeks. Then the embryo is fully developed and the seed can germinate under favorable conditions. Light-sensitive seeds. There are two types of seeds for light sensitivity. Point 1. Positive photoblastic seeds. Seeds of most of the species germinate equally well both in the dark and in light. Therefore, light is not necessary for germination. However, seeds of some species like lettuce remain dormant in the dark. They do not germinate till these are exposed to light. These are called positive photoblastic seeds. Therefore, dark inhibits germination in these seeds. Point 2. Negative photoblastic seeds. Seeds of some species like phlox, Nemophila and Silene remain dormant when exposed to light. They germinate only in the dark. These are called negative photoblastic seeds. Point C. Chilling requirement. Seeds of many temperate trees remain dormant and do not germinate after harvest. Its examples are apple, walnut and pinus. Such seeds need chilling temperature, 1 to 5 degrees, for a few weeks. This requirement is met in nature. These seeds lie buried in the soil during winter. Therefore, they get chilling temperature. But some seeds become dormant in nature. They need artificial chilling. Point D. Dormancy due to growth inhibitory chemicals. There are several naturally occurring compounds which inhibit germination of seeds. Seeds of many species contain several phenolic compounds. These phenolic compounds are ferulic acid, parasorbic acid and coumarin. These compounds are present in the embryo or in the seed coats. They inhibit the germination. Sometimes, an inhibitory hormone abscisic acid prevents germination. Advantages of seed dormancy. Seedling survival. Creation of a seed bank. Synchronized germination ensures a population of plants at same stage of development for outcrossing. Seed dispersal. For example, seed coat scarified by passing through a bird's animal tract. Generation system of seed multiplication. Generation system of seed multiplication is nothing but the production of a particular class of seed from specific class of seed up to certified seed stage. The choice of a proper seed multiplication model is the key to further success of a seed program, which basically depends upon Point A. The rate of genetic deterioration. Point B. Seed multiplication ratio and Point C. Total seed demand. Based on these factors, different seed multiplication models may be derived for each crop, and the seed multiplication agency should decide how quickly the farmers can be supplied with the seed of newly released varieties after the nucleus seed stock has been handed over to the concerned agency so that it may replace the old varieties. In view of the basic factors, The chain of seed multiplication models could be: Point A. 3-generation model. Breeder seed, foundation seed, certified seed. Point B. 4-generation model. Breeder seed, foundation seed, 1 foundation seed, 2 certified seed. Point C. 5-generation model. Breeder seed, foundation seed, I foundation seed, 2 certified seed, I certified seed, 2. Generation system of seed multiplication. Seed is the cheapest and basic input for sustained agricultural production. At the time of release of a variety, small quantity of seed, normally known as nucleus seed, is available with the plant breeder. Commercial quantity of seed is produced after a series of multiplication steps. Starting from maintenance program, in which nucleus seed is multiplied in a generation system of multiplication as breeder, foundation and certified seed. Stages of seed multiplication. After the production of seed, its multiplication for distribution involves following. Nucleus seed. Nucleus seed is the initial amount of pure seed of improved variety or notified variety or parental lines of a hybrid produced under supervision of the evolver of that variety. The nucleus seed generally 100% pure and does not contain other physical impurities. It is produces strictly under isolation to avoid both genetical and physical impurities. Vigor of the original variety or parental line should be retained in nucleus seed. About 500 to 1000 individual plants, IPS, which are healthy and have vigorous growth, are selected from the nucleus bulb plot before onset of flowering. Observations are recorded on each IPS for important and easily observable morphological characters like plant height, stem color, branching. The IPS which are of types or affected from seed-borne diseases are removed. Separate harvesting of the individual IPS is carried out. Seeds are stored in separate cloth bags or paper bags with proper labeling. Breeder seed, stage first and stage second. It is the progeny of nucleus seed. Generally, breeder seed is produced in one stage. But for the reasons like greater demand or low seed multiplication ratio, then breeder seed can be produced in two stages, that is, breeder stage first and second. For stage second, source of seed is stage first. Breeder seed can be produced by original breeder, sponsored plant breeder on agricultural university farms and rarely on government farms. Some of the standards set for breeders' seed are genetic purity 99.9% or more, physical purity 98% or more, germination, as per crop, moisture content less than 12%. Seeds are stored in bags with buff color tags. Foundation seed, stage first and stage second. It is the progeny of breeder seed. Can be produced in two stages with stage first and stage second. Foundation seed is produced on the farms of state agri universities, taluka seed farms, other government farms, state seed corporations and private seed companies. Genetic purity is 99.5% or more. Seeds are stored in bags with white color tags. Registered seed. It is the progeny of foundation seed. It is produced by progressive farmers or registered seed growers according to technical advice and supervision provided by NSC. Registered seeds are genetically pure. Seeds are stored in bags with purple color tags. Certified seed, stage first and stage second. It is the progeny of foundation seed. Genetic purity is 99% or more. Seeds are stored in bags with blue color tags. Truthful seed. It is the category of seed produced by private seed companies and is sold under truthful labels. Companies should maintain field and seed standards suggested for quality seed production as per Seed Act. Seeds are stored in bags with opal green color tags. Differences between certified seed and truthful labeled seed. Certified seed certification is voluntary. Truthful labeled seed. Truthful labeling is compulsory for notified kind of varieties. Certified seed applicable to notified kinds only. Truthful labeled seed applicable to both notified and released varieties. Certified seed, it should satisfy both minimum field and seed standards. Truthful labeled seed tested for physical purity and germination. Certified seed, seed certification officer, seed inspectors can take samples for inspection. Truthful labeled seed, seed inspectors alone can take samples for checking the seed quality.
We politely request, please like, share and subscribe our channel Sri Raj Lakshmi Publication.